3. B cells and their environment. Rather than being passively isolated, a cell interacts with its environment. This section discusses how the environment can affect a cell and how the cell can respond to changes in its environment. 3.4 Objectives. Relate homeostasis to the living condition. Explain optimal range, optimal point, and range of tolerance. Explain the dynamic nature of homeostasis. Describe the effects of hypotonic, hypertonic, and isotonic solutions on a cell. Key terms: homeostasis, isotonic, hypotonic solution, hypertonic solution. 3.4 Homeostasis. Being alive and being surrounded by living things may cause you to lose an appreciation for the narrow range of conditions necessary to maintain the homeostasis, homeostasis, of life. Homeostasis means steady state, but in reality, it is a dynamic equilibrium. The organism is constantly interacting with the surrounding environment to maintain the delicate balance of conditions for life. When the surrounding conditions are altered, the organism must adjust to maintain the homeostasis of life. Optimal point and range of tolerance. Scientists call the precise conditions at which something functions best its optimal point for each condition. The optimal point for temperature in human muscle cells is 37 degrees Celsius, 98.6 degrees Fahrenheit. Most organisms have an optimal range. Within the limitations of the optimal range, an organism's or cell's performance is stable. Most human muscle cells will function equally well in temperatures within a degree or two of 37 degrees Celsius. The optimal range for a muscle cell is quite narrow when compared to the range of temperatures that are optimal for the body. A person can function well and comfortably in rooms with temperatures of 16 to 27 degrees Celsius, 60 to 80 degrees Fahrenheit, depending on the humidity, air currents, clothing, and activities being performed. Throughout this range of temperatures, the body works hard to maintain a constant temperature, homeostasis, for its muscle cells. If the body's temperature increases above the optimal range, a person begins to sweat and his body sends more blood to the arms, legs, and skin. The evaporation of the sweat works to decrease the temperature of the skin and helps cool the blood. If his body's temperature continues to rise, the cells in his body begin to malfunction and he becomes ill. On the other hand, if he becomes chilled, he may begin to shiver, since shivering is the body's attempt to generate heat. To conserve heat, the body will decrease the amount of blood sent to the arms, legs, and skin. If the person is not warmed to the proper temperature, his body begins to stop functioning. The change in temperature forces the body to alter its normal operations to restore homeostasis. The same principle is true for individual cells. The environment affects the functioning of the cell. In the case of temperature, enzymes may be destroyed or rendered non-functional, and cellular processes may occur too fast, too slow, or not at all. If the temperature gets too high or continues for too long, the cell will die. When a cell or organism gets warmer or cooler than the limits of its optimal range, it enters the range of tolerance for temperature. The range of tolerance is that range of temperature in which a cell or organism will remain alive but will not function properly. The further it is outside the optimal range, the more poorly it functions. When it reaches its limit of tolerance, it dies. The range of tolerance for heat and cold for a muscle cell is only a few degrees, quite narrow when compared to the range of tolerance for a skin cell designed to deal with the more extreme temperature fluctuations in its environment. Nerve cells, on the other hand, have a very narrow range of tolerance. It is almost the same as their optimal range. Brain cells are extremely specialized nerve cells and must be kept within their optimal range to maintain proper functioning. Living things were designed with ranges of tolerance that enable them to adapt to their environment. This is why some plants, for example corn, can grow well in moist, fertile soil, while other plants, for example cacti, will not grow well in that same soil, but will grow in arid desert conditions. Some living things are capable of surviving in environments within the optimal range and the range of tolerance for those they display. However, other organisms, for example, humans, cannot survive in environments whose environment normally fluctuates beyond its optimal range to enter a period of dormancy in order to survive unfavorable conditions. The adjustments made by some trees to live through the winter are examples of dormancy. Nonetheless, if the conditions become too harsh and exceed the limits of their tolerance, they will die.
The solutions around cells. Whether a cell is a unicellular organism or is part of a multicellular organism, it is in a solution. One of the most critical influences upon the existence of a cell is the concentration of the substances in the solution around it. The cell membrane is the structure that stands between the surrounding environment and the cell, working to maintain homeostasis. To illustrate this, consider red blood cells and the solution around them. When the concentration of solutes outside the cell is the same as the concentration inside the cell, the solution is said to be isotonic. I-so-tashen-ik. The red blood cells in the bloodstream are a good example of cells in an isotonic solution. The cytoplasm of the red blood cells and the solution that surrounds the cells, plasma, have identical concentrations. Although the solutes in the red cells may be different from those in the plasma, the ratio of water to solutes is the same. Since there is no concentration gradient, there is still movement of water molecules across the semi-permeable membrane, but no net osmosis. If red blood cells are placed into pure water, a concentration gradient is established. When the solution outside the cell has a higher concentration of water molecules and a lower concentration of solutes than the solution inside, the cells are said to be in a hypotonic solution. The solids inside the cell are unable to move through the membrane into the water around the cell. However, water molecules can move through the membrane into the cell. This sets up a one-way flow of water molecules into the cell, causing the cell to swell and eventually burst. The bursting of a cell from internal water pressure is called cytolysis, SICS. If red blood cells are placed in a fluid with a concentration of solutes higher than that inside the cells are in a hypotonic solution. In hypotonic solutions, water molecules will diffuse out of the cell. As the cell loses water, it shrinks. Cells in hypotonic solutions. Since fresh water has fewer solutes than the cytoplasm of a cell, a unicellular organism that lives in fresh water exists in a hypotonic solution. The unicellular and colonial organisms found in such aquatic environments must either deal with the concentration gradient that is around them or die. Many cells that live in aquatic environments have rigid cell walls that prevent cytolysis. As water diffuses into the cell, it fills the central vacuole. As the central vacuole expands, it presses the cytoplasm against the inside of the cell wall, increasing the pressure inside the cell. The turgor pressure inside the cell effectively counters the pressure of the water trying to diffuse into the cell, thus preventing cytolysis. Other organisms that live in hypotonic solutions must have a flexible membrane. Because a rigid cell wall would restrict movement and food-gathering capabilities of protozoans, God designed other mechanisms that enable them to survive a hypotonic environment. Many such organisms have contractile vacuoles. As water molecules diffuse into the cell, they are collected by the contractile vacuole and then released to the environment. If the contractile vacuole cannot establish and maintain equilibrium with the incoming water, cytolysis will eventually occur. Cells in hypotonic solutions. Most cells are not designed to deal with hypotonic solutions as well as they deal with hypotonic solutions. With the exception of cells that live in seawater, most cells do not naturally live in hypotonic environments. Humans, however, occasionally put cells in hypotonic solutions and then wonder why they do not thrive. For example, some gardeners think that if a little is good, then a lot must be better, and they pour more fertilizer on the soil around their plants than the directions recommend. As the fertilizer dissolves, it soaks into the soil. If too much has been applied, the root cells may be surrounded by a hypotonic solution. Ions from the fertilizer enter the cells, and water leaves the cells in an attempt to equalize the concentration gradient. As the water continues to leave the cell, it first loses its turgidity, and then the cell membrane pulls away from the cell wall. This process is called plasmolysis, M-A-H-L-S-I-S, and if it continues, the cell will die. If enough root cells are involved, the entire plant may die. Plant cells are sometimes temporarily short of water. Hot days may cause water to evaporate from leaves more rapidly than the roots can replace it. The results are the same as if a hypotonic solution surrounded the cells. There is a net loss of water from the cells. As the water molecules exit the leaves and enter the atmosphere, the cells lose their turgidity and the plant wilts. Under normal conditions, a cooler evening and night will permit the plant to restore turgidity by obtaining water from the soil faster than it is lost by evaporation. If the wilting continues because of a lack of water or damage to the plant, the cells will experience plasmolysis and die. 3.5 objectives. Compare and contrast passive transport and active transport. Describe the differences between carrier proteins and channel proteins. Distinguish between facilitated diffusion and active transport. Compare and contrast endocytosis and exocytosis. Key terms: passive transport, facilitated diffusion, transport protein. Active transport, endocytosis, phagocytosis. Pinocytosis, exocytosis. 3.5 transportation across the membrane. Most of the previous discussion about substances entering or exiting cells can be explained as simple diffusion through the semi-permeable membrane based on concentration differences inside and outside the cell. Substances such as water, carbon dioxide, oxygen, and many fat-soluble molecules cross the membrane in this manner. However, many substances will not diffuse directly across the membrane. In order to maintain homeostasis, cells must often move substances against the concentration gradient. Passive transport. Many substances cross the cell membrane with no energy expenditure by the cell. Passive transport is the movement of molecules across a membrane with the concentration gradient without the expenditure of chemical energy. The kinetic energy of the molecules, not the cell, supplies the force that drives passive transport. Both diffusion and osmosis are examples of passive transport. Not all molecules, however, are able to diffuse freely across a membrane. The rate of passive transport is influenced both by solute factors and by the particular composition of the membrane. There are several factors besides the concentration of a substance that affect the rate of passive transport across the membrane. One factor is the size of the solute molecule. Some very small molecules, such as water and gases, oxygen, carbon dioxide, and nitrogen, seem to pass easily through almost any cellular membrane. Some other very small molecules and ions pass quite slowly, so there must be other factors besides size. The shape of the molecule will also affect its ability to cross the membrane. Some long, thin molecules pass slowly through a membrane, while round molecules of approximately the same size and mass may not pass at all. The polarity or electrical charges of a molecule also seem to be a factor. Fat-soluble molecules appear to dissolve the phospholipids of a membrane and penetrate more quickly than molecules that are not fat-soluble. In addition to the properties of the solute molecules, the membrane's composition is an important factor in determining the ability and the rate at which a substance will pass through a membrane. Small molecules such as monosaccharides, amino acids, fatty acids, and glycerols penetrate slowly through cell membranes. Larger molecules such as disaccharides and proteins penetrate even more slowly, while lipids barely penetrate membranes at all. Facilitated diffusion. For years, scientists were baffled that some cells permitted the passage of certain molecules, usually specific carbohydrates or amino acids, more readily than others of similar size and shape, even when a favorable concentration gradient exists across the membrane. After much research, scientists determined that a factor contained within the membrane helped or facilitated the passage of certain molecules. Further study revealed that the factors that facilitated the transportation across the membrane were actually proteins that are embedded within the membrane structure. Since this type of diffusion requires a facilitator or helper, it is termed facilitated diffusion, and the embedded proteins are called transport proteins. Scientists believe that there are two types of transport proteins involved in facilitated diffusion. Both are highly specific, allowing only certain substances to cross the membrane. The first is called a channel protein and forms tiny pores or holes in the membrane through which specific substances pass. The other transport protein is termed a carrier protein. The carrier protein has a specific receptor site that binds to the molecule it is designed to carry. As it binds to the substance, the carrier protein changes shape and the molecule passes through the membrane. Once the molecule has passed through the membrane, the carrier protein reverts to its original shape. Active transport. Many times the movement of molecules or solutes is against the concentration gradient, from an area of lower concentration to an area of higher concentration. Just as in facilitated diffusion, carrier proteins are utilized to help the molecule cross the membrane. However, the difference is that energy is required. When energy is required and the substance is moved across the membrane against the concentration gradient, the process is called active transport. Some types of active transport systems are termed pumps because they use cellular energy to pump substances against their concentration gradient. In active transport, the substance that is to be transported binds to a carrier protein. Once that occurs, the carrier protein uses cellular energy to change its shape, and the molecule is transported across the membrane. Once the molecule is across the membrane, the carrier protein returns to its original shape. The processes by which cells store and use energy are discussed in the next chapter. Bulk transport, endocytosis and exocytosis. Large substances like starch and protein molecules are too big for any of the transport systems studied thus far. Some cells are able to eat large molecules and other cells whole. There are other mechanisms that cells use to transport substances in bulk across their membranes, endocytosis and exocytosis, both of which require energy and so are types of active transport. Endocytosis, ENDOH-CYTOH-CIS, is the process a cell uses to transport substances in bulk across the membrane and into the cell. When a cell contacts the substance to be transported, the cell membrane folds inward until it completely surrounds the substance. Once the substance is completely surrounded, the membrane is pinched off into the inside of the cell. There are two types of endocytosis, depending on the kind of substance that is transported. The term phagocytosis, phag, ou scatohs, referring to the process of a cell ingesting another cell, applies to the movement of any bulk solid material across the membrane. Pinocytosis, pin-o-cy-to-to-cis, is the movement of bulk fluids or solutes across the membrane. Sometimes phagocytosis is called cellular eating, and pinocytosis is called cellular drinking. The reverse of endocytosis is exocytosis, exocytohs, and is the process whereby vesicles or vacuoles in the cytoplasm fuse with the cell membrane and release their contents into the solution outside the cell. Cells use exocytosis to secrete materials produced by the cell, such as hormones or proteins, or to release non-soluble, indigestible wastes from food or phagocytic vacuoles.